All right, last couple of slides here. Last couple of things we need to talk about. So we're going to go back. Let's kind of jump back a little bit now to the brain and some of the cerebral spinal fluid. So we're going to take a look at kind of cerebral spinal fluid, how it's made, and where it it comes from. Right. So it's created in what are called the ventricles. The ventricles are basically these cavities or open spaces, right, that make the CSF, the cerebral spinal fluid. So if we were to just take a quick look at the brain that we've cut, sliced frontally, right, so this cavity here, so this is actually this hollowed out area. So in the cadaver, right, we see that's going to be basically an open hole, open space. But in real life, right, in a real functioning brain, all of this will be filled with our cerebral spinal fluid. And so we have in the brain 4 ventricles. Right, so our 4 ventricles are the lateral ventricle. So we have 2 of them, right? So we have 2 lateral ventricles. They're both just called the lateral ventricle. You can see, so they're like this horn-shaped structure here. And this is the ventricle that you see in that photo when we're looking at the frontal view. So we're seeing the piece of the ventricles, the lateral ventricle here, and then of the other opening. Right? And we can see from the side, so it's all of this. You can see there's both sides there. Right? It contains all of this component here. So it loops around. Right? So all of that here is going to be the lateral ventricle. So those are our ventricles 1 and 2. Right. Finally, underneath that, we have the 3rd ventricle. Right. So lateral. The 3rd ventricle is, that's not that. Right. This guy here. And finally, we have the 4th ventricle, right, which is this one here. So you can see the 3rd ventricle, it's fairly wide when we're looking anterior to posterior, yet mediolaterally, right, quite narrow. And then we have the opposite effect with the 4th ventricle, which is quite narrow along our sagittal view, and then wide here in the frontal plane. Right, so these are hollow spaces that contain our cerebral spinal fluid and make it. Right, so I'm just kind of explain a little bit of how the CSF will move through the ventricles into the spinal cord. And then around the brain as well. Okay, so let's say we're starting in the lateral ventricle, right? So maybe I'm just going to start this over a bit here. Get rid of the super messy ones. All right, so we've made our cerebral spinal fluid here within the lateral ventricle, right? And so the flow is going to come down from the lateral ventricle into the 3rd ventricle through what's called the interventricular foramen, right? So interventricular, between the ventricles, and foramen is the hole. So it's the hole between the ventricles, right? And that is at that juncture right there. We can even see it here, right, on the frontal view. So it's going to connect not only the lateral ventricle to the third ventricle, but also the two sides. So our left and right lateral ventricles will connect through the interventricular foramen. Right? Once in the third vesicle, it will flow down through what's called the cerebral aqueduct. So getting a little more fancy with our terminology, I suppose. Right? So the cerebral aqueduct is going to be the passage between the third and fourth ventricle. And then from the fourth ventricle, it basically vents out into the subarachnoid space through what are called apertures. Right? So our apertures are basically these little, almost like little holes, little vents, almost like a sieve, I suppose, that allows for flow into that subarachnoid space, which then travels through the rest of the brain and spinal cord. The last thing here is the central canal, and so the central canal basically just extends from the fourth ventricle and travels all the way down through the middle of the spinal cord. So we go back, we actually see it here. So there's a little hole right in the middle of the spinal cord, which is the central canal, and so that is actually filled. That little gap there runs all the way through, right, and is filled with cerebrospinal fluid. So it's encased here between the pia and the arachnoid mater. So within this layer here, we have cerebrospinal fluid, and as well a little bit on the interior there. So it creates a kind of that encompassing effect there. Okay, and there's again, the ventricles from the frontal view, right? You can see when they're cut out there, that would be filled with that cerebrospinal fluid. And so our last kind of thing we're going to cover here is the formation. So how we make cerebrospinal fluid. So we're going to have to go back a little bit to the first set of slides from the nervous system and have a look at those ependymal cells. They are basically the epithelial layering that we saw within the central nervous system. So the non-neuronal, the non-neuron cells. Remember, we had the astrocytes, oligodendrocytes, and then the ependymal cells were part of that grouping as well. But basically, so that cerebrospinal fluid, the CSF, right, is made by what's called a choroid plexus, right? And so a choroid plexus is made up of kind of two components here. So it's basically a capillary, right? So we have a capillary running here, which is a blood supply, right? And these ependymal cells. Remember, there's the cilia-laced cells, and this would be the outside. So this is that cavity of the ventricle. So this is facing the hollow space. So if we were to go back here, right? If this were expanded out here, right? This would be inside the ventricle. So it would almost be like we're looking at this bottom part here, right? So if we looked at that there, we could have that teardrop choroid plexus. And so actually, well, it would be the top part, because in our photo, this is the cavity of the ventricle. So it would be basically lining around the inside of the ventricle there, right? So that's the inside of that of the ventricle on the outside of our photo here. So basically what happens is as plasma from the capillary it filters into the ependymal cells, right? So we have a nutrients being filtered out. So the plasma comes through the capillary, right, into the ependymal cells that basically add the fluid to it, right? So we have a bunch of like glucose, oxygen, some ions, proteins, right? So don't need to know all that detail about what's in it, but so these nutrients, these components get pulled from the plasma of the capillary into the ependymal cells, which add their fluid to it and then secrete them into the cavity of the ventricle. And it's these cilia, right? So these projections, the cilia here, they're going to wave around and help circulate the cerebral spinal fluid. So it's going to help it move around. So we talk about how it moves through the ventricles, right? It's going to be propelled and circulated by the cilia of these ependymal cells. And so there's three kind of key, I guess, functions. Two are physiological functions. One is like more of a medical function of cerebral spinal fluid. So the first one is protection, basically. So the cerebral spinal fluid, right, we know it's that layer, so it sits between the brain and the skull, essentially. And it provides that extra bit of cushioning between the soft tissue of our brain and that harder tissue of our skull. So for an example of how this would work, if you were to just slap your hand down on the table, right, you would, depending on how hard you hit, you might hurt your hand, but you definitely would feel it a little bit more in your hand, as opposed to if you were in a swimming pool, right, and you try to hit the wall with your hand through a swimming pool. So just that extra bit of basically fluid resistance, right, it slows your hand down, so the impact on the wall is much less. And so the cerebral spinal fluid basically slows down your brain before it slams into your skull, in the case of kind of any trauma or accident or incidents where that would happen. Right, so it provides a layer of protection. The second key feature of cerebral spinal fluid is buoyancy, right? So it actually helps make things a little bit lighter inside the brain, right? So it also, it makes things a little bit lighter. So we think about the buoyancy. So basically, it allows your brain to float within your skull, right? And so it's not there's not as much pressure, there's not as much weight being applied downward, so that the ability to float helps make things just a little bit lighter and therefore just easier from a movement sort of standpoint. And finally, the third sort of function of cerebral spinal fluid, so it's not quite a physiological function, but I'm saying it's more of a kind of a medical function. It can be used as a marker of health for the nervous system. You might have heard of the term a spinal tap before. That's where they basically a needle is put into your not quite your spinal cord, right? So it's actually placed below that L2 level. So when the spinal cord proper actually ends, but right beneath that, in that pool of cerebral spinal fluid, right, you can collect a sample of it there from what's called a spinal tap, and you can analyze it, knowing that it's contains all these different nutrients and proteins, etc. So you can actually use it as a marker of health of the nervous system. Right, so cerebrospinal fluid, three main functions: protection, buoyancy, and that marker of health of the nervous system. Okay, and so that brings us to the end of this video, and we will continue on with the peripheral nervous system, looking more at the autonomic nervous system, as well as the sympathetic and parasympathetic in our next bit of lectures. So thanks for watching, and I'll see you soon.